So I figured I'd put this up with the basics of COVID-19. I'm Professor Michael McDarby. Been teaching biology for better than 30 years. Background training in parasite biology, not viruses, but also did work on evolution of diseases, and something like this, evolution becomes a really, really big deal. Just want to talk a little bit about how the virus works and how we work in response to the virus, and what the implications are for the pandemic to some extent. Virus. You'll hear people talking about killing the virus, and then you'll hear experts go, well, you can't really kill a virus because it's not really alive. Is that viruses don't fit some of the nice, neat things we have on a list for things that should be alive. Is that when they're flying around in between people, they really have no capacity to react to the environment except to bump into the right cell and connect. That's it. Living things interact with their environment. They don't grow. They're made fully formed. They do reproduce like living things. They do evolve like living things, but they do break a couple of the rules. So a room full of biologists, maybe half will go, yeah, they're living, and half will go, no, they're not. So it's not even to us. Doesn't matter to them. Anyway, what you've got is a casing with these, what are called spike proteins. You may have heard people talking about spike proteins and other proteins sticking out through a covering that is stolen from the cell that made them. So they have a membrane casing, which is not true for a lot of viruses, but it also makes them a lot more vulnerable to the detergents in soap. So when they talk about washing your hands with soap, then the soap turns out to be a pretty important component of that. And you can disrupt the membrane. How quickly they lose their ability to infect people once they're free and dried out, nobody's quite figured out yet. How many viruses you need to pick up. For an awful lot of diseases, there's what they call a viral load. You need to be exposed to a certain threshold of virus numbers before they can really set up shop in you. And nobody's really sure what that is for COVID-19. They spread in respiratory droplets. How big are the respiratory droplets? Are they tiny enough to float in the air for long periods of time? Are they big enough that they sink fairly quickly? Are there viruses in both of those, and you need the big ones in order to pick up enough viruses? Those are all things that are not as easy to answer as some of the other things we know about coronaviruses. So it's not that easy for us to really tell you those things. Odds are they're going to be passed in fairly large respiratory droplets. So the stuff that spews out of your mouth when you talk, or even when you breathe to some extent, breathe on a cold winter day, you can see the stuff coming out. But that stuff has some weight to it, and it tends to drop. So the 6-foot bubble, it doesn't tend to get beyond the 6-foot bubble. Also, if you got a barrier in front of your face, even if it forces the air out through the edges, you go, the air's going in and out anyway. Well, yeah, but if it goes out here, it doesn't get very far from you, and it's dropping right around you. So your infective bubble, if you're carrying the virus, is a lot smaller with the mask. Is that you're basically dropping them on your shirt. So that's one of the reasons why a lot of people are going, wear the mask, wear the mask, wear the masks. And it's kind of, I don't need to explain the science to you. It's just common sense that's what would happen to stuff that you're breathing out if these things were trapped in the little particles of spittle that are coming out of your mouth, that they're not going to get very far from you if they have to work their way out around the edges of a mask. And a bunch of them stick to the mask as well, so they don't even get out. They talk about, we can find the virus here, we can find the virus there, we can find the virus on surfaces after a certain amount of time, we can find it coming out the other end of your body. It's way easier to find evidence of the viruses. They test for molecules that are actually inside the virus. That doesn't tell you whether these are infective or not. Just because we can find them doesn't mean that the viruses that we're finding are viruses that could make somebody sick. And that's a lot tougher to figure out. So the evidence on that is a lot less clear. Welcome to science, is that we learn a lot of stuff, but there's a lot of stuff we don't know, and there's a lot of questions that we kind of go, well, we think this is what's happening, but we're not 100% sure. Let's look for something else. And then we build up evidence. The evidence for the masks, there wasn't a lot of study on anything other than surgical masks coming into this pandemic. And what there was, pretty crappy studies. And so people go, oh, the studies go to say the masks don't work. And then people quickly put together studies, and those were pretty crappy studies. I haven't seen a really good study on the effectiveness of non-surgical masks, except for just the common sense variety of, watch the air come out of somebody's mouth without the masks and with the masks, and imagine viral particles in that. You won't necessarily have to see, strict evidence. You have a molecule on a whole bunch of cells in your body, the linings of your breathing system for one, that these spike proteins attach to. Viruses need a way in, and they generally do that by popping onto a molecule on your cells, interacting with the molecule and getting their stuff inside. Doesn't necessarily have to be the whole virus, because the virus is carrying a payload of code. Most viruses carry DNA, which is the same code that we use to make all the proteins that do all the stuff in our bodies. When you make proteins, though, we use a molecule called RNA as kind of the actual making the protein code. And RNA is what's in these viruses. There's a number of viruses that have RNA in them, and they work in various ways. So this one has RNA in it. It's got a protein to kind of help jumpstart what's going on. It does a very odd thing where it feeds the RNA through the protein making system. And one of the first things it makes is another protein that kind of sits there and watches the string of proteins come out. And it goes, okay, that one's done, snip. And then it forms a functional protein. Okay, that one's done, snip. And it forms another functional protein. And it's making the building blocks to make more viruses, and the building blocks to make more RNA to put in more viruses. And then they get up inside the surface of the cell and manage to get themselves out. They get into the mucus of your breathing tubes, and that's how you spread them. Now, one thing that's weird about this virus, that's weird about just as a pathogen, typically when you're sick, some of your symptoms have to do with you're trying to actually fight whatever it is that's making you sick. You're on a fever, you're trying to change the temperature. Different chemicals work differently at different temperatures. A fever kind of helps some of your immune reactions and hurts some of the stuff that's going on for these guys. It often doesn't work, right? We still run fevers for the flu. It doesn't really do us any good. However, you get the flu when you cough. You can't clear flu viruses out of you with a cough. What that does is it helps the viruses to spread. A lot of pathogens give you symptoms that will help their little offspring get out of your body into the next person. Because if you're a pathogen, that's your whole life. Get into a host, reproduce, get your offspring into another host. Because it's very unlikely you're going to be able to spend the long haul in the host, because the host will eventually come up with ways to get rid of you. We spread the virus with no symptoms at all. That's very unusual, especially for something that has just moved from an animal of some sort into people. Usually a jump like that, the pathogen is not that compatible with the new host, and it tends to screw us up in a bunch of ways, including producing a lot of legitimate sick symptoms, which it is doing. But how, the idea that it can spread before you ever have the symptoms that would normally spread it is very, very unusual for one of these new pathogens. There are a few mutants that they've detected. The mutants that they've been looking at are the ones that change the shape of this attachment protein, the spike protein. Nobody really knows what that means in terms of whether it makes it more infective, because the spike has to attach to the protein, what they call the ACE receptor on your cells. And say a mutation makes it attach tighter. That may not actually help the virus get in. That may actually be a bad thing for the virus. There may be a certain level of looseness that it needs in order to do what it needs to do. One of the problems that you have with any virus is that, what do we come up with treatments? If you got the virus, how do we deal with it? And we try to do that the way any pathogen gets into you. What is the pathogen doing chemically that we can poison? We can screw up and kill the pathogen. And it'd be really good if the chemical we give it to screw up the pathogen doesn't screw up chemistry in you and hurt you. The virus uses your machinery to make proteins. You don't want to screw that up. You have proteins being made all over your body. You do not want to get into the middle of that machinery and screw with it. So an awful lot of therapeutics for viruses, you have to hope that the virus is doing something that your cells just don't do. And sometimes to get out of the cells, like with chickenpox viruses and other herpes viruses, there's chemistry involved in getting the viruses out of the cells, and a Tamiflu against influenza goes after that. Some of the treatments for herpes viruses go after that. But there's a very small list of chemicals that you can go after. And then you got to get the chemical to the target. And most of what these guys are doing is happening inside your cells. Not that easy to get to that. And once they're free of the cells, they're not really doing anything, just looking to bump into the next host. So there's not a lot of chemistry you can poison. The best you can do is, with soap, you can make the casing fall apart. That's good enough. So therapeutics, a lot of the stuff they're testing, and they go, oh, we'll come up with something and you could treat it. Think about all the viral diseases that we have. We don't have any real treatments for it. And we've been working on that for a really, really long time. We can slow them down. We can reduce your symptoms, but we can't really, as they say, kill the viruses because we don't have the right targets for it. So this gets into your body. And you have cells that are gonna touch on this and go, well, this doesn't belong here. And it literally will pull molecules out of the surface of this thing and hand them to another cell. And that's how your antibodies get made. You have another cell that'll take this molecule, give it to it just the right way, and it will make a molecule that'll fit on there like a little cap. That's what an antibody does. Fits on there. Once it's on the other molecule, which happens when you get exposed to more viruses, the other end basically goes, hey, immune system, come in here, do something about this. And your immune system kind of descends on what's been flagged. And that's how, when you have antibodies to a virus, you can't get enough of them can't get in and actually infect you before your immune system has been flagged. You've been flagged with the antibodies, and you're coming in and you're going after them. That isn't how we fight viruses off typically, though, because it takes a while to make antibodies. The antibodies are good for the next infection, to keep you from getting sick again with the same pathogen. What we do is we have detection systems for just viruses that go after that, just kind of a general, And being general, it's not that great. It can, in some people, really help lower the ability of the virus to take hold. We also have detection systems inside our cells for virus invasion. We have detection systems for cells that are infected with virus. A big player in that system is something called interferon. And one of the probably big players in this epidemic is interferon, in that, I had the flu vaccine back in November. You get a vaccine, it exposes you to molecules that are like the molecules on, in this case, the flu, and you make antibodies to attach to it, to whatever flus they think are going to be around. It's always a guessing game, so sometimes they guess right on what flus are going to be around, sometimes they don't. Right now there's a flu brewing in China that may be out next in a couple of months. The vaccines are already set and being made for this year's flu season. So if a new one pops up and starts to spread, the flu vaccine will protect you from the ones they figured were going to be around, but not the new ones. Anyway, but you make antibodies eventually to all sorts of things on these molecules. But you also release interferon to help clear the initial infection. If you have, okay, back up. This coronavirus does another weird thing. One of the first things it does when it starts to work in your cells is it sends out a chemical that blocks your ability to make interferon. So your body goes, oh crap, we got virus infected cells, we gotta make interferon, and the chemical that has been released goes, no you don't. And so that's why it messes with your immune system in weird ways. It's also possibly why if you get really, really sick with it, eventually your immune system hits a point where it isn't shutting it off, and it goes, and it just comes in and it produces what's called the cytokine storm, which can actually kill you. Is people who are dying are to some extent dying from their own immune system overreacting. That's why the anti-inflammatory that works on very, very late stage patients, anti-inflammation is an immune system reaction. Those kind of work. I like to explain things. Here's what you may have. If you've been exposed to other viruses recently, if you've been exposed to a viral-based vaccine like the flu shot, then you already have interferon in your system. And that may be enough to keep coronavirus from doing anything particularly nasty to you. I had COVID very, very mildly. I believe it's because I had the flu vaccine fairly recently and I still had enough interferon that it kept it from setting up shop. When they look at groups that should be getting sick because they have comorbidities and stuff, like homeless people, people that are packed into tight places like in prisons, nobody's looked at daycare workers, but I would be curious to see that. People, if you're in a situation where you're getting exposed to diseases, especially to viruses all the time, then you may be protected because your interferon levels may be up enough that it can't really get in you and set up shop. As you get older, your interferon levels are not as reliable, so that's another reason why old people are more susceptible, even though we've been exposed to a lot of diseases. And it isn't just exposed, it's exposed recently. You kind of need that interferon response to kind of be ramped up a little bit in order for it to work. And I have been doing studies on throwing interferon at people early on, but the only study I know of, I was following it, and they went, well, we were going to do this, and then our partner that makes the interferon for us went out of business, so we're not sure what the heck we're going to do. Welcome to the wonderful world of science. So you make antibodies. Now, for the most part, they're trying to produce vaccines to this particular molecule. But if you get exposed to these viruses, there's other molecules sticking out of this casing that you're going to make antibodies to. Some of those molecules are very, very similar from this coronavirus to the other coronaviruses. And there was a study just out this past week, the middle of July, where instead of looking at antibodies, they were looking at the cells that kind of remember how the antibodies need to be made, your T cells, which are a better measure of immunity. And actually, for people that hear, oh, your antibodies are waning, you're losing levels of antibodies, your T cells are staying fine. You're going to stay immune for a good long time to this thing. The people that had it in 2003 had the first version, the SARS version. They're still immune to it. But there's a whole bunch of coronaviruses that cause colds. Most colds caused by rhinoviruses, but there's some that are caused by coronaviruses. There's a strong indication that people that have been exposed to those and have antibodies to some of these other molecules can't completely keep it from setting up shop in them, but they can control it enough that they get no or very minimal symptoms, because they're not clearing the viruses completely as if they maybe had a vaccination for this, but they are clearing it out. And so that's important to know that people go, oh, it doesn't affect everybody the same way. Well, no, it doesn't, and we're starting to figure out why. But it turns out there's a whole bunch of factors, and you can't always be predicting just exactly what your level of protection is. And it does hurt people of all ages, with little kids. It gets into other parts of your body. You have these molecules that it attaches to everywhere. And in little kids that have leakier systems, apparently it has a possibility of getting into the brain and doing nasty things there. That's still up in the air, not entirely determined. But, and you know, there's young people that are even people that don't have other kinds of issues, that, you know, they just pick the wrong day for their immune system to be sleeping, and they wind up very, very seriously ill, sometimes dead. There was the kid in the news just recently who went to a COVID party and, you know, dying in the hospital, went, well, I didn't think it was a serious thing. I believe, I'm looking at my notes here to make sure that I wanted to cover everything. I believe that covers the basics of coronavirus and COVID-19 for this particular pandemic as of the middle of July. Things are changing every single day. Anybody who has, even if you don't have a huge background in science, but you're interested in really getting a good way to track what's happening in a very readable form, although it does get somewhat technical. There's a blog out of the American Association of Science called Into the Pipeline by Derek Lowe. I have no connection to this. I just read it every day or every time that he puts an entry out, and very, very informative. Keeps track of what's happening in all the vaccines. He works in the development of pharmaceuticals, so he's very familiar with how the system works. And if you got extra time, is that you can go into his archives and read, there's a little area that's things I won't work with. And that's usually very, very entertaining. Anyway, and my website's Prof McDarby, but my stuff is all for my classes, so I'm not gonna probably have much of anything else up. So that's it for the coronavirus.